Hi guys, I am back again for Dr. C's class so we can do the next phase, which is meiosis. Again, I got a crash course, so I'm trying to remember like something I learned like 25 years ago. Again, there's going to be lots of gaps. Your job is to fill in all the gaps. I'm just here to help you with the hands-on part of it, okay? So, what you're going to do right now is you're going to make salt dough again. Okay, so if you need to pause me, go ahead and pause me. I'll hang out here while you go make the salt dough and then you can come back. Push play. Alright, so go ahead and do that. Okay, so here's what we're going to do. Just like we did in the other mitosis, you're going to take your salt dough and I'm going to go ahead and tilt my camera down so you can see our work surface. Same spot again. Okay. Sharpies. We need again 3 colors. Take your salt dough. We're going to go ahead and flatten it out. Oh, I forgot my pizza cutter. Okay, you need something, pizza cutter, serrated edge, whatever. Alright, so flatten it out so it'll be easier for us to draw our chromosomes and chromatids, and honestly, it's a lot of math, okay? It's like 23 and 40s, well, I don't know. We're going to end up with cells with 23 chromosomes or chromatids, right? Okay, all I know is to go from 46 up to 90 something and we come back down. So, again, your job is to fill in all those blanks. But, I do know there are 2 phases, right? We're going to do the first phase right now. During that first phase, they get friendly with each other and they cross over, right? So what we're going to do is we're actually going to draw our chromosomes after they have crossed over and hung out with each other, right? Because then they pick up DNA from each other. So I am going to start with blue, and we're not going to draw all those chromosomes because that's a lot and that's going to take forever. We're going to start out with 3, okay? So, and just like before, they're going to all line up in the middle, but they're going to have pairs on them, right? So, I'm going to start with 1, 2, 3, right? And I think I'm going to do green for my other side. Right, they line up in pairs. Okay, but they've crossed over, they've hung out, they've gotten friendly, whatever you want to say it. So I'm going to add on one side, I'm going to have an extra little green part to it. So that means I'm going to have to do the same thing on this side where he's going to have an extra little green, red, oh my goodness, blue part on this end, right? Just like that. And because, again, more science words, I know that there is a center. I'm going to make my center pink just so I can remember where I started out. And I know the center's got a name and I can't remember it. Again, that's your job, not mine. Okay, so when we get done, should look something like that, right? Because in one of the phases, they've already hung out, they've got DNA from each other, and they've lined up in the middle. So what happens? Now the cell splits. And just like in mitosis, we're going to very carefully score right down the middle, right? Because they have those little spindle looking things that are going to pull it apart. So we're going to pull very, oh, I didn't do it enough. We're going to pull very carefully. And we should end up now with 2 cells, okay? So just like before, I'm going to move these chromosomes over towards the middle, and all I'm doing is kind of just patting it down and bringing it back around again, trying to keep them in the middle and making it round. Okay, it may take some time, and I might need to adjust my little chromosomes because they went a little funky, but that's okay. That is easy to do. That is why we use Sharpie. Okay, so once you have your 2 cells, that ends the first phase. See, I can learn. All right, that's the end of the first prophase. I think that's what it's called. But don't quote me on it. I don't want to get you in trouble. Okay, so we have a relatively somewhat circly shaped cells again. All right, and I'm going to mark my chromosomes again just so it'll be easier for me to remember where they are, because I have one that went kind of crazy over here. Okay, and that went that way. Okay, I'm just touching them up, the ones that need to be touched up, just so it's easier for me to see them for our next part, right? That one really went funky. I need to put that center back on. He really went crazy. Okay, so in the beginning of, this one looks better, in the beginning of the second prophase, they don't have to hang out because they already have DNA, right? So what we're going to do now, same thing. They're going to kind of line up in the middle, which I kind of have, this one's actually easier. They're kind of lined up in the middle. And they're going to split again, right? So this time we're going to split our chromosomes from the chromatids, just like we did in mitosis. And kind of where I've got it, it's kind of a little funky, but you can make it work. And now I'm going to split it again. Remember, they're going to split right down the middle after they've lined up. And voila. Again, I'm going to move him over towards the center. So that way, I now have funky little chromatids. Squish it down in the center, right? And if I were to just draw it again, I just follow my line and I can see where my pinks were, the pink centers. And voila, there is one cell. And you do that for each one, so that way by the time we get done, you should have, how many cells? How many cells should we have? You should have 4. And each one should have 23 chromosomes. I'm going to put my centers back on. Alright, this one's a little messier. But you guys get the idea, right? You're going to do that and you're going to end up with 4 individual cells. All right, hopefully you do good and you can fill in all the blanks that I obviously left out, because guess what, I'm not a scientist. All right, good job guys. Hopefully this helps, and I will see you guys next time.